All right, we have just gone through the longest mini lecture. This next one might be the second longest, but we're going to be able to talk about, finish up the majority of chapter 16 here. And then we are going to move on to other smaller topics, okay? So let's finish up chapter 16. We've talked about the overview of it. We haven't talked about how you regulate it, which means how you add more to it when it's running down, when you need more intermediates, and how you turn it on and off. One of the ways to turn it on is to replenish it. So this really is the third topic that we always talk about, and we're going to talk a little bit about anabolism because we always have to do that, right? Now, if you look at this, here's a big chapter that shows citric acid cycle, and there's 4 red arrows on this. These are called the anaplerotic reactions that you want to talk about how they replenish the citric acid cycle. So look at those 4 reactions and just see where they go in. You replenish the citric acid cycle in front of the top of the cycle. On the left side of the cycle, where you add oxaloacetate for 3 reactions, and then you add malate. But I believe you only do the malate if you're a plant. Okay? There's one of these that's most important to us, but look at what you're doing in all of these cases. They all have reactants that have 3 carbons, either pyruvate or phosphoenol pyruvate. And you're building up the pyruvate into a 4-carbon molecule, usually oxaloacetate. So there's actually 3 different ways to make oxaloacetate. 1 way to make it from pyruvate, 2 ways to make it from phosphoenol pyruvate, which is 1 step above it in glycolysis. And also, remember how energetic that acyl phosphate is. It's hugely energetic to break that. It's a $60 bill. So here you're using a $60 bill to make a 4-carbon molecule from your 3-carbon molecule, rather than to get energy out of it. Okay? You're kind of not spending the money, or you're spending it not to burn stuff, but to build it. So if you look at these, you have the malate one is the weird one that is important to plants, and I believe we might have one little homework problem about it. To us animals, what's really important is the pyruvate carboxylase. And by the way, sometimes I say these things the wrong way. They have similar names, so be clear that if you're reading it, you know it's right. If I'm saying it, sometimes I get it wrong. But right now, it's pyruvate carboxylase is the important one that takes pyruvate and makes oxaloacetate out of it. The other thing you can do is you can take the energetic molecule PEP and also use that. You'll notice that there's one reaction that's in blue that could be from glycolysis. They're not showing it on here for clarity. But if you notice that in glycolysis, normally what you do is you take the PEP and you turn it into pyruvate. So normally we have that arrow going on. We're just not showing it because this is for showing the citric acid cycle. It's not for showing glycolysis. Okay? So realize that all of these intermediates can take part in anabolism, and that's why sometimes instead of burning the carbon by putting it through acetyl-CoA, you actually want to build the carbon by putting it through oxaloacetate. You do 1 step of building before you even put it into the cycle, and then you use the cycle to make more, say, alpha-ketoglutarate from the oxaloacetate. Or also maybe your oxaloacetate concentrations have just dropped too low. Maybe you need more oxaloacetate to be able to make more acetate into CO2. You might need to turn the cycle more. So you might need to make more CO2 on the one hand. Either way, you want to make more oxaloacetate, right? So this is how you can do that. It costs energy, but you can also build stuff out of it. If you're putting oxaloacetate into the cycle, that means you can also take alpha-ketoglutarate out of the cycle without losing too much later. And then you can turn the alpha-ketoglutarate into glutamate. Remember that we talked about that. And then the glutamate can be turned into glutamine, proline, arginine. Look at all the things in the outer ring of blue that you can make from alpha-ketoglutarate on the right, or oxaloacetate on the left, or citrate on the right. You can make fatty acids out of that, or succinyl-CoA. You can actually use that to make your heme groups. A lot of those topics are going to be later in the course when we talk about anabolism. We just want you to know right now, oh, sometimes I actually want more alpha-ketoglutarate, and in that case, I actually want to turn some of my pyruvate into the 4-carbon oxaloacetate. Building, not burning, is the main concept. So those are the 4 red arrows that you have here. Notice that 2 of them start with pyruvate, 2 of them start with phosphoenol pyruvate, but the ones that start with phosphoenol pyruvate tell you that they are PEP enzymes. You see how they have that in there? And phosphoenol pyruvate can actually work with bicarb, or it can work with CO2, but it requires a GDP. It actually takes a phosphate and puts it onto a GTP. That's really cool reaction. You just need to know that it exists, but we aren't going to focus on that particular one because it's a very specialized reaction like the heart does. The main one that is most important to your body takes place in the liver and kidney, and it's the top one, which is pyruvate carboxylase. It's the only one that is the main one that makes pyruvate into oxaloacetate for your body. You can also take pyruvate and you can do a similar thing to turn it into malate, but that's malic enzyme, and it's more widely distributed. It's more of a bacterial kind of function or a plant function. What animals do is we do pyruvate carboxylase, and I'm going to talk about that turns pyruvate, and it's always a question of where do you get your carbon from. It gets its carbon from bicarb, not directly from CO2. So you're not fixing CO2 gas. You're actually reacting bicarb, which is already dissolved form of CO2. But it costs an ATP. So this is the important thing. It does really cool chemistry, consumes 1 ATP, and the important thing about this is you don't turn on this enzyme unless you have a lot of acetyl-CoA around. This is generally what you do when you need to make more, you need to burn more acetyl-CoA. If your if your citric acid cycle is chugging along and burning as fast as it can, and yet you're breaking down carbons and producing more 2-carbon units than the citric acid cycle can handle. How do you make the citric acid cycle turbocharged? How do you make it turn faster? Well, you turn some of the pyruvate into oxaloacetate using this enzyme. So you only want that to happen if you have a glut of acetyl-CoA. If you're starving for acetyl-CoA, you don't want to take the pyruvate and turn some of it, take it away from acetyl-CoA. So realize that logic. Acetyl-CoA accumulates. You want to do more citric acid cycle. You do that by turning on this enzyme. So that means acetyl-CoA is an important green triangle for turning pyruvate carboxylase on. If you have a lot of acetyl-CoA, you can get rid of it by burning more, and it's kind of like increasing the fire. It's kind of like poking the fireplace to increase the combustion of the acetyl-CoA by having more oxaloacetate. So how does the chemistry actually work? I'm actually covering up the middle steps here deliberately because I only want you to look at the beginning and the end. The beginning is in the upper left corner, and you see that you have bicarbonate, which is able to attack ATP. You burn an ATP in doing this, but you make a nice acyl phosphate. This carboxyphosphate that you have at the top is an activated, you see how it's an anhydride form of the bonds, 2 double bonds separated by 1 oxygen. And so this is very reactive, and this can actually phosphorylate the biotin in the middle. And that's the other thing I want you to see. I want you to see that weird shape. We don't have any amino acids that look like that, so you might be starting to think vitamin. We also have a long chain that looks like lipoate. You might be thinking that there's some parallel there. And so you have this covalently attached to the enzyme. It's a biotinyl lysine group. So it's attached through an amide bond to the lysine. And then you basically put the phosphate, or I'm sorry, I said phosphate, but I meant you put the carbon onto it. Because you see over here, you have the carboxylated form of biotin. That's what this group can do for you. You can carboxylate it, which means you now have an activated 1-carbon unit that wants to react with something else. So you react it with pyruvate. You do the enol chemistry. You basically do you turn it into the reactive enol form, and then you react the enol form of it with the C, so that you form a CC bond, and you make oxaloacetate. So realize that the pyruvate enolate form is very crucial to this because it makes the CH3 into an active chemical agent. So biotin's really a CO2 carrier on a long flexible arm. When CO2 is bound to it, it is actually activated so it can go on to something else. You just move it to the place where it needs to be, and it lets go of it, and it makes oxaloacetate. So it gets the CO2 from the bicarbonate, it has one site where it picks up, where it basically picks up the CO2 with the help of hydrolyzing an ATP. Then it goes over to site 2 where the pyruvate's waiting, and it reacts with the enolate form of the pyruvate to make the oxaloacetate. ATP hydrolysis is essential for this. You're not going to get anywhere because biotin can't react with bicarb by itself. It needs energetic help, and it goes through the carboxyphosphate. And now you can form the CC bond on the reactive enolate. You know, it requires a lot of help for this thing to go forward. CC bonds are not easy to make. You're not a plant. But this is one way in which you can mimic a plant because you can form a CC bond using biotin, but you need the biotin for it. So the electron pushing, don't worry about it. When we get to the cofactors, I want you to recognize the shape, but not necessarily be able to do all the electron pushing. You can study that another time. So this is why biotin is actually, it's something that you need to make or you need to eat. And so it's abundant in many foods because this function of moving one carbon units around is very useful. So a lot of bacteria make it. I think I'm not sure if plants make it, but I wouldn't be surprised if they do. And it's classified technically as vitamin H, but it's part of the B complex of vitamins. But honestly, it's in so many places. As long as you're eating food, usually you're getting enough biotin. Okay. The one thing that can happen is not getting enough biotin, but eating too much of the thing that binds biotin. Because things need biotin, they make a protein that binds biotin very tightly. If you eat too much of that protein, you can have enough biotin, but it could be covered up by this biotin binding protein, which is called an avidin because it is so avid in binding. So egg whites contain a lot of protein called avidin, and you actually do have to be careful if you're eating a huge ton of egg whites. You know, they have the avidin, which is supposed to defend against bacteria growing by covering up all the biotin. But it might defend against you growing if you have so many egg white binding proteins, so much avidin in your system that all your biotin is covered up and can't be used by the rest of your body. And so this trivia question, which Disney villain is at risk of a biotin deficiency? Not from not making it, but from eating too many egg whites. Okay? I wish we were in person because I would like to know if somebody gets this, but if you know the songs by the great Howard Ashman, one of my literary heroes, Howard Ashman wrote for Gaston, I eat, oh my goodness, students can usually quote it for me, and now I'm blanking on it. You can go look it up. So listen to Gaston, and he eats like eggs, right? Because he's a bodybuilder. He's trying to get the protein up, but the problem is, if you're eating raw eggs, the folded avidin will get into your system. Now, if Gaston wasn't eating raw eggs, he was just eating cooked eggs, no one who eats cooked eggs has this problem. Why would cooked eggs never result in biotin deficiency, no matter how many you eat? Think about it. What is avidin? Avidin is a protein. What happens to proteins in eggs when you cook them? They unfold, they lose their activity. So by cooking eggs, you are automatically avoiding biotin deficiency problems. So that's why I'd recommend to Gaston, but of course he probably wouldn't listen to me because he's too busy listening to all the people who tell him what he wants to hear. Okay, so the thing is, we want to look back and we want to see these three biological tethers. So we had lipoate and biotin in this unit. And just notice what the shapes are like. These are always covalently bound. We're later going to have an acyl carrier protein that is a long chain. And again, the details of what the chain looks like, you can look that up. What you care about is what's on the end of the chain. And on the end of the chain, you have disulfide for lipoate. You have this nitrogen pentagon for biotin that can bind carboxyl groups. And then we have another one that's a thiol. And that's a little bit like how we were talking about CoA as well. We'll talk about that one later. The only thing is that you might see pantothenate this other weird structure that you need for this other weird enzyme, that's going to be a vitamin 2. And it turns out that that is vitamin B5. So the final thing to talk about is regulation. And this is where we have a lot of red and green figures. I want you to see the main story of these figures. So rather than memorizing each of these regulation points, I want you to see that you have a lot of the right side of the citric acid cycle and pyruvate dehydrogenase, because dehydrogenase is what feeds the right side of the cycle by making acetyl-CoA. If you look at the right side of the cycle, you have most of those steps. You have 1, 2, 3, 4. Just 4 steps, including steps 1 through 3, and also pyruvate dehydrogenase. 3 of those 4 steps are upregulated by calcium. So I want you to know that general trend. Calcium will tend to upregulate these right side, these catabolic enzymes. And that makes sense, right? Muscles are contracting, they're using calcium, and the calcium will turn on the citric acid cycle in order to be able to keep contracting. It does aerobic metabolism in that case. So the other thing that you can look is you can see which one of these are regulated. Step pyruvate dehydrogenase, which is sort of step 0, I suppose. But you also have steps 1 and steps 1, 2, and 3. The 2 decarboxylations are also highly regulated. And so you just want to say, oh, those are the points where you have lots of regulation. The bigger enzymes also tend to have more binding sites for regulators, which also makes them highly regulated. So I want you to follow this pattern. You want to be turning on and off. The on-off switches are located on the right side, and they're also located before you get to the cycle. Just like with glycolysis, the steps 1 and 3 were the highly regulated. Again, steps 1 and 3 are the most highly regulated, and also step 2 and step 0 in this case. And if you look at the delta G's for those, the delta G's for those all tend to be negative. They all tend to drive forward because you are getting rid of CO2, which is very energetically helpful, things like that. These are really driven forward. So look at the other trends. When you have excess ADP or AMP, that means you need more ATP. So if you have empty ATP boxes that have been broken open, those will turn up these enzymes. Don't worry about AMP versus ADP. We're not getting to that level of detail. We just want to know in general, if you need energy, AMP is going to be high relative to ATP, and that's going to turn on these enzymes in general. They're downregulated by ATP or NADH. So if you have enough phosphates, if you have enough electron boxes, you're actually going to have red X's with those. And again, the same pattern is what you want to look at. There's one other thing that's kind of cool. Remember, by the way, that we had this fructose, we said that citrate would back-regulate glycolysis. So this is actually the step in glycolysis. The citrate comes back and it downregulates. If you have a lot of citrate around, that means that this particular enzyme doesn't need to break down the glucose as much. So that's an explanation for what we already talked about. And that's this general rule. If you have a lot of citric acid intermediates, then you actually probably want to slow down the waterfall of carbon that's coming into the top of the citric acid cycle. You probably want to turn down glycolysis. So they are connected in that way. The other thing is that we've already talked about how sometimes the enzymes get together. There's steps 8 through 2 actually associate in this case. So malate dehydrogenase, citrate synthase, and aconitase all get together, and they actually form a big group. They form a union that they bind to each other so that you don't lose the oxaloacetate. This is how oxaloacetate can be taken away as soon as it's made. You actually make a little funnel for it, so as soon as it's made, it goes right into the citrate synthase. It never really sees the concentration in the exterior of the cell. And in fact, you can even find this channel. Remember that all these intermediates are negatively charged. Oxaloacetate has 2 negative charges on it. So what you want to do is, if you have the structure of one of these things and you find the active sites, you want to look for a connecting path that is all blue, because it's all positive amino acids that will help the oxaloacetate move from the step 8 binding site to the step 1 binding site, where it can start another turn of the cycle. And that's what you see right here. You see that little blue path? That's what oxaloacetate will follow, and then it will make citrate. And that's on the left side of this metabolon. So realize that metabolons are really just multimers. They're just aggregates, but they're ordered aggregates of the enzymes. So you're bringing them together. You're doing the handoff thing rather than throwing a pass. And that actually ends up our chapter 16.